Number 1. Some students investigated the effect of drinking caffeine on reaction time. They used a drink containing 32.25 milligrams of caffeine per 100 centimeters cubed. This is the method used. 1. Divide the students into 4 groups, A, B, C and D. Measure and record the reaction time of each student using the ruler drop test. Students in group A drink 200 centimeter cubed of water. B, 200 centimeters cubed of the caffeine drink. C, drink 400 centimeters cubed of the caffeine drink. And group D drink 600 centimeters cubed of the caffeine drink. Repeat step 2 after 15 minutes. Describe how to do the ruler drop test. So you need to hold a ruler just above the open hand of the other student so that the 0 is just above their finger. Let's say, make sure it's the same each time. Drop the ruler and the other student catches it as soon as possible. And then you need to record where the ruler is caught, meaning the distance on the ruler where the top of the finger is. Table 1 shows the mass of the caffeine taken in by each student. So there we've got our group and our mass of caffeine in milligrams. Calculate the value of X. Well, in order to do that, we're going to need to look back at what the volume of these drinks are. And I'm just going to look back and then write them on the table here. 0 mass of caffeine in 200 centimeters cubed of water, as you would expect. And then group B, they consume 200 centimeters cubed of caffeine. C, 400 centimeters cubed, and D, 600 centimeters cubed. We know that the value of X is 600 centimeters cubed, so we're going to have to add 200 and 400 together. So in other words, we're going to have to add B and C together. So that's 64.5 plus 129, which gives us 193.5. Why did group A drink water instead of the caffeine drink? Well, we need to be able to compare the effect of no caffeine with the effect of caffeine, so it's used as a control. Table 2 was used to convert the results of the ruler drop test into reaction times. Estimate the reaction time for a student who recorded a distance of 23 centimetres. So if we have a look at the distances, 23 is between 22 and 24. So we would expect the reaction time to be between 0.212 and 0.221. So 217, or a range of 0.215 to 0.218 is acceptable. Students calculated the decrease in their reaction time after the drink compared with before the drink. Figure 1 shows the results for each student. We've got the groups along the x-axis. Group A, remember, have no caffeine, and Group D have the most caffeine. We've got decrease in reaction time on the y-axis. Describe the effect of the mass of caffeine taken in on the decrease in reaction time. So we look at that general trend. It is an upward trend, isn't it? As the mass of caffeine increases, the decrease in reaction time increases, which sounds a bit confusing, but we're literally just taking that off of the graph. For 3 students, the decrease in reaction time was negative. Give the reason why the value was negative. Well, if their reaction time was greater after they actually had the drink, then they're actually slower. What is the range of results for Group C? So we need to go to the graph. The lowest value of the decrease in reaction time is 0.010 seconds. Then the highest value is 0.180. Suggest 2 variables that should have been controlled in this investigation. There are lots you could choose for, so I'm going to go for the starting point of the ruler needs to be placed above the hand and use the same hand as well. And then you could have written students need to be of the same age, same gender, height, having had the same amount of sleep, etc. But make sure you only write 2 and 1 on each line. Explain why the ruler drop test does not involve a reflex action. Well, that is because catching the ruler is a voluntary action, so it's not automatic. It involves the conscious or the thinking part of the brain. There has been a rapid increase in the percentage of carbon dioxide in the atmosphere since 1960. Carbon dioxide is a greenhouse gas that contributes to global warming. Name one other greenhouse gas. Methane. Global warming causes climate change. Give two effects of climate change. Again, you could have lots of different examples here. So I'm going to go for rising sea levels, making sure I've got one on that line. And then my second one, I'm going to put change in species distribution. But you could also put ice caps melting, desertification, extreme weather, change in migration patterns, loss of biodiversity, coral bleaching, crop failure, etc. Just make sure you only give two and make sure there's only one on each line. Plants take in carbon dioxide from the atmosphere, and figure 2 shows part of the carbon cycle. So we've got our carbon dioxide in the atmosphere, and we've got microorganisms that are carrying out respiration, so they're going to add carbon dioxide to the atmosphere. We've got green plants carrying out respiration, so they're going to be adding carbon dioxide to the atmosphere. Animals eating the carbon compounds of the green plants. And then animals carrying out aerobic respiration and therefore releasing the carbon from those compounds out as carbon dioxide. Any animal waste will be decayed, and that involves microorganisms, and they'll have to respire. And of course, green plants, when they die or bits of leaves drop off them, decay is going to happen there, which also involves microorganisms and therefore respiration. So we use this diagram here and pop that into our 6 marker with all the writing. We'll start with photosynthesis. Carbon dioxide diffuses through the stomata into the leaf. Carbon dioxide and water are both needed for photosynthesis to take place and for glucose to be produced. Some of this glucose will be used by the plant for respiration, meaning that the carbon from this glucose will be released from the plant as carbon dioxide. The glucose made in photosynthesis can also be converted into other carbon compounds such as amino acids, which are obviously going to make up proteins, lipids, and they can also be stored as starch. And these organic carbon-containing compounds can then be consumed or eaten by animals. And then these animals will use these carbon compounds to release energy through respiration. And then because aerobic respiration is happening, that means that carbon will be released into the atmosphere in carbon dioxide. And then carbon compounds will also make up the animal's biomass. Microorganisms can feed off or digest decaying plant or animal waste. These microorganisms will help to return the minerals from the waste into the soil, but they will also be carrying out respiration as well. So that means some of the carbon they have absorbed will actually be released into the atmosphere through respiration as well. Question 3. Vectors are used in the process of genetic engineering. Which 2 statements are correct? Vectors are enzymes used to cut open the DNA molecule. No, they're not. Vectors are used to insert genes into cells. Yes. Vectors are used to isolate the required gene. No. Vectors are used to stimulate cell division. No. Vectors are usually plasmids or viruses. Yes. Now, these vectors that are used in genetic engineering are obviously different from the kinds of vectors we talk about in communicable diseases. There's lots of different types of vectors. Scientists have genetically engineered a variety of wheat to be resistant to herbicides. Herbicides are chemicals that kill plants. The herbicide-resistant variety of wheat, so those that won't be killed by the herbicide, will give a higher yield, so there'll be more of it, than the non-herbicide-resistant variety. Explain why. Well, the non-herbicide-resistant plants will be killed by the herbicide, and the herbicide-resistant wheat plant won't be killed by the herbicide. So there will be less competition for light, water, minerals, etc., because the non-herbicide-resistant plants have been killed. Right? So that means there's going to be more light for our resistant plants. More light, more photosynthesis, and therefore more glucose can be made, and then that can be converted into carbon compounds for the plant, and then the plant's able to grow. That means it's going to give it more yield if it's growing. Give 2 examples of genetic engineering in use today. Do not refer to herbicide resistance in your answer. Okay, so remember the technique again. 2 examples, 1 on each line. So I'm going to choose production of human insulin as my first one, and crops that are resistant to pathogens, therefore disease. That's a pretty handy one. But you could also put things like resistance to frost, plants with bigger fruits, improved taste, added nutrients such as the vitamin A in the golden rice, for example. Scientists working on the Human Genome Project have now mapped the entire genetic code of humans. Explain one way this could be important for people in the future. Genes that are linked to disease can be identified, leading to better prevention or treatment of that disease. Animals have adaptations to survive in their environment. These adaptations may be structural, behavioural, or functional. Draw one line from each animal adaptation to the type of adaptation it is. Male palm cockatoos use sticks to beat on hollow branches to attract females. So that's definitely behavioural. The harmless hornet moth has black and yellow stripes to look like a bee or a wasp. Well, that's structural, so you can see it. It's what the organism looks like. And we have sea spiders, which have automatic muscle contractions that move oxygen around their bodies. Well, that is definitely a functional kind of physiological adaptation there. Plants also have adaptations. There are more than 28,000 known species of orchid plants. Many orchid plants grow attached to other types of plants, have brightly coloured flowers, produce large quantities of pollen, and produce thousands of tiny light seeds. Describe how these adaptations help orchid plants to survive and compete. Growing attached to other plants is going to help them to be supported by the plant they're growing on, and that's also going to help them to obtain the water and the minerals from that plant. Then the bright colours will attract pollinators such as insects to transfer the pollen from one flower to another, so we can have fertilisation going on there. Large quantities of pollen increase the chances of fertilisation and reproduction. And if we've got tiny light seeds, then that means they're going to be more likely to spread over wider areas. It's going to be easier for them. And because there are thousands of them, it means there's more chance that some are going to survive. A rare orchid has been found in the mountains in China. The orchid has pale yellow flowers. DNA analysis of the genome shows that it is an ancestral species. All other present-day orchids evolved from this ancestral species millions of years ago. One present-day species has bright purple flowers. Describe how an orchid with bright purple flowers may have evolved from the ancestral species which has pale yellow flowers. Within a species, there is genetic variation due to sexual reproduction and or mutations. These genetic differences may have given rise to a new purple flower colour. Now, if the purple flowers are better suited to a particular environment or a change in the environment, then they're going to be more likely to survive and reproduce. And therefore, the allele that causes the purple colour will be passed on to their offspring. And then over many generations, the purple flowered orchid may be so genetically different that it can no longer breed successfully with that ancestral orchid, and therefore leading to this new species of orchid. The DNA code determines the sequence of amino acids which are joined together to form a specific protein. Table 3 shows part of the amino acid sequence for the colour pigment protein in 5 orchid species. The rest of the amino acid sequence is the same for all the species. So we've got our table here, species A to D, ancestral, flower colour, amino acid sequence, so we've got a key as well. Suggest which orchid species is most closely related to the ancestral species. Give a reason for your answer. So let's have a look at the amino acid sequence of the ancestral species. Basically, we've got to find the one that is most similar to that. And as I look down the rows, my eyes are drawn to species D. And if we look ALA, yes. LU, yes. GLY, yes. ISOLU, TYR, yeah. But we've got a difference here with this amino acid, and then all the other amino acids are the same. If you look above at A, B, and C, there are more differences between those and the ancestral species. So D is the species, and it's because only one amino acid is different. Question 5. Some students estimated the population of daisies in a school field. This is the method used. 1. Find a place where some daisies are growing, put the quadrat down, count and record the number of daisies in the quadrat, repeat steps 1 to 4 at 4 different places in the field, calculate the mean number of daisies per quadrat and use the data to estimate the total number of daisies in the field. Now, which two improvements would increase the validity of this method? So it means that it's a decent investigation that you can have meaningful results with. So what are our choices here? Do not put any quadrats near the trees. Repeat for another 10 quadrats. Yes, I definitely would do this one because they're only doing it 4 times. So you want to increase the number of quadrats there to make this more of a valid investigation, definitely. Use a long tape measure. I don't see how that's going to really help with this particular investigation. Use a random method to place the quadrats. Yes, absolutely, because we need to eliminate the bias. You can't just go, oh, there's loads of daisies growing over there, pop the quadrat down and then choose another spot where there's loads of daisies, which is what the method suggests that students are doing. So we need to randomise it and so that we can get a more representative sample of the field area. Because of course in biology we need to sample it. We couldn't possibly count every single daisy in that field. So we need to take a representative sample and then estimate a total number. With an improved method, the students calculated the mean number of daisy plants to be 7.65 per quadrat. The students used a quadrat measuring 50 centimetres by 50 centimetres, and figure 3 shows the school site and the dimensions of the school field. Okay, we've got a bit of a funny shape here to work with. Calculate the population of daisy plants on the school field and give your answer in standard form to 2 significant figures. So first of all, we need to work out what the area of the field is. And if you have a look at this shape here, the best thing we can do is divide it into a rectangle. And then we know we've got 62 metres times 164. So that gives us 10,168. Then we're going to need to add that to the triangle we're left with. And we know it's half base times height for the area of a triangle. So that the base is going to be 164 metres divided by 2. So we've got half of that. And then we need to times it by the height. And how we're going to work out the height is we've got 130 metres in total minus 62 metres. So we're left with 68. So that height is 68 metres. Anyway, yeah, so then when we do the half base times height, that gives us 5,576. So we add those two small areas together to give us the total area, which is 15,744 metres squared. The students said that their mean number of daisy plants per quadrat was 7.65 and they only did 4 quadrats. So we need to do the total area, 15,744 times the 4 quadrats times the mean number of daisies per quadrat, and that gives us 481,766.4, and we need to put that in standard form to 2 significant figures, so that is going to be 4.8 times 10 to the 5. The students noticed a very uneven distribution of daisy plants in the field. Explain how different biotic and abiotic factors could have caused an uneven distribution of daisy plants. Remembering biotic factors are living factors, so that might be other plants or humans or animals, and then abiotic are non-living factors, so for plants, things that affect it like light intensity, ions in the soil, water availability, pH, pollutants from a nearby road, for example. So if we have a look back at the diagram, We can see that we've got trees, sports pitches, school buildings and a road. So really we need to comment on these things in our answer. The trees will reduce the light available for photosynthesis, so there may be fewer daisies in this area. The trees will also absorb water and mineral ions from the soil, so that could reduce the number of daisies because they'd have fewer minerals and less water. There will be a lot of trampling on the sports pitches, people playing around their games, and that could kill the daisies. Other plants growing in the field will compete, so this is biotic factor. They're going to compete with the daisies for the water and the mineral ions, so that means that daisies will get less and they won't grow as well. You've also got the school building there, and that might create some shade over certain areas of the field, and that would mean less photosynthesis for the daisies. The daisies also might be sensitive to the pollution from the vehicles on the road, and this might reduce the daisy growth as well. Last question, 6. This question is about homeostasis. Define the term homeostasis. So your AQA definition that they like is the maintenance of the internal conditions of a cell or body for optimum cell or enzyme activity. Name the hormone released if blood glucose concentration falls too low. That is glucagon. And on this occasion, they will only accept the correct spelling for this because it's so similar to glycogen. A lot of the time with spellings, they do accept phonetically correct spelling. So you're okay most of the time, but certain words like glucagon, you've got to get them right. 2 people were sent to a hospital to find out if they have diabetes. This is the method used at the hospital. Do not eat or drink after midnight. This is called fasting. Measure blood glucose concentration at 9:30 a.m. Drink a glucose solution at 10 a.m. and measure the blood glucose concentration for the next 3 hours. And we have got the results here. We've also got a little key which tells us what the normal blood concentration should be when you're fasting, so you haven't eaten for a while, and then when you have eaten. Person A and person B both have diabetes. Describe how figure 4 shows that person B has diabetes. Use the data from figure 4. On this occasion, because both person A and B have diabetes, we're not comparing B with A because they're both diabetic. So we have to compare person B with what the normal figures should be. So if we have a look, B's fasting blood glucose was 8.2 millimoles per decimeter cubed. So before they've even eaten anything, it's much higher than the normal range, and the normal range is 3.5 to 5.5 millimoles per decimeter cubed. Then, after the glucose drink, which they have at 10 o'clock, their blood glucose increases to 13.2 millimoles per decimeter cubed. So we're bringing the data off of that graph there. And that is much higher than the normal non-fasting range of 4 to 7.8 millimoles per decimeter cubed. I could have written that in there as well, you could do. And their blood glucose decreases very slowly to 12 millimoles per decimeter cubed, which is much higher than a normal range should be. So even after 3 hours, it doesn't even return back to their starting glucose concentration. Person A and person B had a test to measure the concentration of insulin in their blood when they were fasting. Table 4 shows the result and suggests which type of diabetes person A and person B have. Give a reason for each answer. Person A, look, they're actually making a lot of insulin. So this must be type 2 diabetes because they can still make lots of insulin, but their body cells can't respond to it, or their cells have become resistant to insulin. Notice I've underlined the word cells because it's really important that you write cells rather than the body, otherwise you won't get the mark for that. Person B has got below the normal range of insulin production, so we'd say they were type 1 because they're not producing enough insulin. 6.5 Toxic hypoglycemia syndrome, THS, has caused the deaths of hundreds of starving children in some tropical countries. The starving children have had nothing to eat all day. The starving children then eat many lychee fruits. The lychee fruits contain a molecule which stops an enzyme in their liver from working. This enzyme normally converts stored fats into glucose. Children who have eaten during the day are not affected by eating the lychee fruits. Starving children may die from eating many lychee fruits, but children who have eaten during the day are not affected. Explain why. I think this is quite a hard question because this relies on you remembering that the glycogen stores in the liver have to be converted to glucose so that people can respire. So if you haven't eaten for a while, the glucagon converts glycogen back into glucose. If you remember that, and you've got to remember all of that to get to this. Starving children will have used up all of their glycogen stores. So they're going to need to get their glucose from stored fat. But if the starving children then eat the lychee fruit, then they won't be able to convert their stored fats into glucose. So this means they'll have no glucose available to carry out respiration, so they'll get no energy, and if they don't get any more food, they could die. Children that are not starving, they will have glycogen stores in their liver, so they don't have to rely on the fats being converted to glucose to survive. Their glucagon will continue to convert their glycogen into glucose, so they'll get energy from respiration with that glucose. That's quite a hard question. Anyway, you made it to the end. Well done. Thank you for listening. Good luck in your exams. See you in the next video.